is very important because these two receptors are now our models, or some of our models, for doing behavior. And what you see is that at lower concentrations, phenylethylamine is preferentially activated by phenylethylamine, TAR4 is preferentially activated by phenylethylamine, and TAR3 is preferentially activated by isopentylamine and cyclohexylamine. One of the most interesting observations here is that if you look at the dose-response relationship for TAR4 and phenylethylamine, TAR4 is by far the most sensitive main olfactory receptor that we know of. So you can see the thresholds that we measured for phenylethylamine in this assay are down in the 10 to the minus 14th molar range, which is many, many orders of magnitude lower than for any other odorant receptor that's been studied. Even the sensitivity to N-methylpiperidine and cyclohexylamine are those affinities are relatively high. If you were to compare this, for example, to TAR3, you can see that TAR3 is relatively sensitive to isopentylamine and cyclohexylamine, but the sensitivity of TAR4 to its best ligand is much higher. So the question is, do you also see this breadth of tuning in this odorant response sensitivity in vivo? To do that, we can cross our genetically targeted strains to our reporter, and we can look in vivo and ask whether the response profiles that we see are valid when you stimulate the animal not in the aqueous phase, but in the vapor phase. We're getting closer and closer towards behavior here. We want to be able to know what these inputs are doing because we want to be able to do behavior. So what we do is we can take these animals and put them underneath a standard CCD camera, thin the bone over the olfactory bulbs, and image from the glomeruli directly. That's how we get the images, the movies that I showed you before. When you do this, the kinds of images that you get look like this, right? So here is the red image showing the positions of TAR4 glomeruli in vivo. This is a anesthetized, freely breathing animal. This is the resting fluorescence from the indicator, in this case synaptofluorin. And synaptofluorin increases its fluorescence when the glomeruli are activated. And so you see, if you put on phenylethylamine, you can see the individual arrows show the positions of TAR4 glomeruli. You can see the TAR4 glomeruli are activated by phenylethylamine. They're activated a little bit by trimethylamine, not so much in this case by isopentylamine, and also you get activation with cadaverine. So the breadth of response that we see in our in vitro assay is also seen in vivo. In addition, the sensitivity can also be seen. So here's the TAR4 glomerulus, it's this one here. And in fact, at low concentrations, when you put on phenylethylamine, amazingly, the TAR4 glomerulus is the only one that we see come on. And I hope you appreciate that in the olfactory literature, that is an unprecedented, relatively unprecedented thing, to be able to put on an odorant and activate a single glomerulus, even restricted to the dorsal bulb, is a significant advance. Because now you have a target that you can go record and know that you're activating a particular receptor and a particular glomerulus in a reliable way. If you look across the amines, this is a max projection of responses to a couple of amines, what you see is that glomeruli in this region are preferentially activated. And what we see is that that region corresponds to the other TAR glomeruli. So we have a very nice handle. We have a set of glomeruli that we know we can activate in vivo. We have cross-reactivity just like normal ORs. We have preferred ligands, and we can now look to see how individual TAR genes contribute to whatever behavior we're interested in. And the first behavior that we started to look at was prompted by previous work that had shown that the animals show seemingly innate responses to amines. And what do I mean by that? So Steve Lieberlis had shown that phenylethylamine, this compound that activates TAR4, is enriched in the urine of carnivores, in carnivorous cats, and mice seem to avoid this chemical without training. And so they're averse to phenylethylamine. And so one of the questions we wanted to address first was whether the TARs are required for mice to exhibit that aversion. And so what we did, without going into too many details, we used a method called transallelic recombination in mice to delete the entire cluster of tar genes, leaving tar1 intact. So we got rid of the tars, made a tarless animal, at least removing the olfactory tars. And the first thing that I'll show you is that the responses that you get to amines at low concentrations in this region of the dorsal olfactory bulb, in the deletion animals, those responses are gone. And if you put on amine concentrations, you can activate other glomeruli. And the responses to other chemicals don't seem to be affected. I want to make sure that we all understand that when I'm showing you these imaging experiments, we're only imaging from the dorsal olfactory bulb. There are many hundreds of glomeruli that we are not addressing and that we don't have optical access to. And so in order to figure out, it's very difficult to get a handle on whether the amines are activating other ventral glomeruli. There could be 10 or 100 glomeruli that are also activated by phenylethylamine. We just don't see them. Instead of trying to image from the ventral bulb, we decided to go ahead and do our experiments because we figured that the behavior would tell us whether we had an input that was significant for the detection of phenylethylamine or not. So the first question is, well, do the tars mediate this aversive response to amines? And to test that, we did an assay that was very similar to what other groups had done. You take a clean cage, you put a naive mouse into this cage. This animal is naive at least to the testing conditions. And you put an odor source in a compartment. And it's simply a place preference assay. You ask how much time the animal spends in the odorized chamber when the odor is an odor or if the odor is some control like water. We video track the animals over a 3-minute period, and we can then come up with an aversion index, which is the time that they spend in the chamber when the chamber has an odor minus the time they spend when the chamber has a control, water. Right? So a negative value will mean that the animal is averse to the stimulus. Does it work? Yes, it works. If you look at control odor, for example, TMT, which is known to be aversive, it's aversive to mice, it's aversive to graduate students. At high concentrations and even low concentrations, we see aversion. And what I'm plotting here are data for 3 genotypes. Wild-type animals and litter mates that are homozygous cluster deletion, they don't have tars, or heterozygous cluster deletion, they have half the complement of tars. All right, so the assay seems to work. What happens with the amines? So the first thing that we were able to show was that if you put on phenylethylamine at relatively low concentrations, you see a tar-dependent aversion. That is to say, animals that have tars show aversion. In the knockout animals, the aversion seems to be removed. It is not only phenylethylamine that shows this. We see it for isopentylamine and methylpiperidine and cadaverine. I should mention the diamines have different, I don't know if anyone's ever smelled these. These things smell horrendous, by the way. They smell like rotting dead fish. Cadaverine's not so bad despite its name, but the idea is that these, at least to humans, have very different qualities, but in fact the animals avoid them similarly, and these are all tar dependent. In addition, if you use the urine of a carnivorous cat, particularly in this case a puma from the Philadelphia Zoo, what you see is that animals normally avoid that stimulus, but the knockout animals don't. So the tars seem to be necessary for animals to exhibit this aversion. So that's in animals in which we've removed the entire tar repertoire. Is it possible that if we remove a single tar that we would be able to see such changes? So what we did was we went back to our tar4 deletion animals, right? So now tar4 is gone, but the rest of the tars remain. If we do our in vivo imaging assay, we can see that in fact we've had an effect. So here's phenylethylamine response in a region of the bulb where the tar4 glomerulus is, in an animal in which tar4 is tagged. If you look in that same region, which is actually tagged, but in fact it's homozygous deletion, you don't get a response to phenylethylamine, but you do get responses to isopentylamine and methylpiperidine, and the response to puma urine, the volatiles from puma urine are not seen. If you look at the behavior, you see something that parallels that. So if you knock out tar4, we see a loss of aversion to phenylethylamine, but the aversion to isopentylamine and N-methylpyridine is still intact. The response to, in this case, 2 different predator urines is also abolished. Okay. So one possibility is, well, okay, you knock out the TARs, maybe the animals just don't smell the amines anymore. And so this is something that we are still very interested in addressing. And I think our data suggests that is not the explanation for what's going on, and I'll tell you why. So first of all, what we spent a lot of time in the last few years doing is addressing this question by thresholding animals. So what we can do is we use, again, a head-fixed go-no-go paradigm, and we basically puff on odors in a very controlled way, and we ask the animals if they can smell them. We lower the concentration. We can put together a psychometric curve looking, that tells us something about. If you look at these are data for phenylethylamine concentration in wild-type animals and cluster deletion littermates, so these are animals that are lacking all TARs, what you see is that there is a significant decrease in sensitivity between the 2 genotypes. You even see this if you look at TAR4 deletions by itself, although the effect is not as large, right? In TAR4 animals, you can also see a deficit in detection of phenylethylamine. So I should mention, though, these animals are clearly not anosmic. They can still detect phenylethylamine, and they can even detect it at relatively low concentrations. I should mention that the lowest concentrations, in fact, all the concentrations that are listed here, I can't smell it, right? So we're thresholding animals for stimuli that we can't even smell. So the question really is, well, are the animals detecting, let's say, phenylethylamine under conditions in which they're failing to avoid? Okay, how do we test that? So what we can do is we can actually take animals and we can do an aversive conditioning experiment to ask them if they can smell phenylethylamine under conditions which they don't normally avoid. So what we do is we take mice of different genotypes, we expose them to phenylethylamine, they smell the odor, and then we immediately pair that with either a saline injection as a control or lithium chloride to make them sick. They pick up an aversion then to phenylethylamine. We can then test them later in our place preference assay to ask whether or not we've induced aversion. And what we find is that animals that are deletions which normally don't show aversion can be made to avoid the same concentration of phenylethylamine by lithium chloride injection. So when you give lithium chloride, what happens is that the deletion TAR4 deletion animals begin to both detect and avoid phenylethylamine. So the idea is that our TAR4 deletion animals, at least, are failing to avoid phenylethylamine at a concentration that they can detect. Whether or not there is a component of this aversiveness of the odor that's intensity dependent is unknown. It's a very difficult thing to work out, but we plan to do that in the coming years. Okay? So just to summarize, what my lab has been able to show in the last few years is that a majority of the TARs are mapped to a specific region of the olfactory bulb, and that these dorsal glomeruli are preferentially responsive to amines. And TAR4 is very sensitive, even among the TARs. But interestingly, we can show that not only TAR4, but the other TARs as well, seem to contribute significantly to determining the behavioral sensitivity of mice to these chemicals. In the field of olfaction, that is a significant advance. It's the first time that we can take main olfactory GPCRs, remove them, and have an effect on, in this case, sensory detection, right? Stimulus detection. Okay. So these days, so we're continuing to ask a number of related questions. And so one of the intriguing questions here is, to what degree is the aversion that we see to all of these amines related to perhaps this domain structure? That is to say, to what degree is the aversion learned, or to what degree is it perhaps hardwired? Is there this idea of whether there are there's innate valence in the olfactory system is a contentious issue in the field. In gustation, it's pretty commonly accepted that certain tastes are appetitive and other tastes are not, right? They're disgusting, they're aversive. In olfaction, that's kind of a contentious issue. A very simple-minded hypothesis might be that selective activation of glomeruli in this region, maybe by amines, elicits a response in this region, and this region may be perhaps a quote-unquote labeled line that tends to give aversion. The alternate idea would be it doesn't really matter what the input pattern is. This organization is not so important. The animals are somewhere in their life associating the presence of amines with some aversive cue, stimulus, and as a result, the amines take on the aversive quality, take on this aversive valence. We think that we have the tools to begin to address this, at least at the circuit level, by rewiring the inputs to the olfactory bulb and by using selective activation of glomeruli using channel rhodopsin. So the idea is that if this structure is important, if activation of this region means aversion to the animal, we should be able to elicit that in a number of different ways. So the first way that you should be able to do it is using channel rhodopsin. We have animals in which TAR4 expresses channel rhodopsin. That would let us use light to activate that region, the TAR region. We also have animals in which M72, the first strain that I showed you, express channel rhodopsin. These would be glomeruli outside of that region. And so the idea is that we could go in with a laser and we could activate these glomeruli in different animals and then look at whether or not that has an effect on whether they want to be in a particular part of this chamber in a place preference assay. So these are ongoing experiments. Currently doing them. I don't have an answer. Preliminary data are that if you activate these glomeruli, you can elicit aversion. But of course, the control will be to show that activating other glomeruli does not elicit aversion, and so we're doing that. Okay, so this is one area that we're following up. The related question would be, well, you don't have to do this using light. You can actually do this using odors as well. So here is our map of our olfactory bulb. Genetically, we can treat this like a patch panel. We can pull out one input here and plug it in somewhere else. So how do we do that? So let's say we have an input domain, right? So for example, TAR4, here's the TAR4 gene in cartoon. Here's its corresponding glomeruli in the animal in this D3 region, this blue region. If we take the TAR4 coding sequence, the part that codes for the protein, and we stick that gene into the M72 locus, the M72 is normally found out here, what you end up producing is an animal that has two TAR4 glomeruli, the native one in its normal position and an ectopic one in a new location. This animal is interesting in and of itself. You can do some interesting experiments with it, but what we can also do is cross this with the TAR4 deletion, remove the endogenous glomerulus, and now we have a glomerulus that we can address specifically, we think, with low concentrations of phenylethylamine, but now it's in a new location. And so the idea would be to ask whether these animals are of phenylethylamine or not. And again, I don't have an answer for you. This is an experiment that we're doing. These animals are being generated, and we should have them hopefully within the year. Okay. Another direction that we're going in is prompted by an observation from another lab, and it's a pretty interesting one. So again, Steve Lieberlus, who works on TARs, showed that TAR5 which we know is activated by trimethylamine, which really smells horrendous. TAR5 is enriched in mouse urine, and what he's reported is that mice are attracted to trimethylamine. So on the surface, this would tend to argue against the idea that input to the TAR domain necessarily gives you aversion, or it might argue for something such as the idea that this was all learned, or it could be that this is modified by experience. What my postdoc, Adam Dewan, noticed in their study was that their animals were individually housed, and as a result, it's likely that many of them were dominant, and that they were males, they're dominant males. And so we've started to look at this issue using a number of assays to ask whether or not dominance has an effect on perception. So the dominant animal will push the other animal out of the tube. He's still okay. He's going to pop up. Okay, he's all right. So this is one of the assays that's been classically used to measure dominance in mice. The other is scent marking. So if you take two mice, you pair them for a little while, they will establish dominance. If you put them in a cage in which they're separated but they have odor contact with each other, they can smell each other, what will happen is that the dominant male will scent mark like this, and as my postdoc says, the subordinate will pee in the corner. And so this is sort of the best data that you get from that assay, but this is sort of what it looks like. And preliminary data we have suggests that in fact the response to trimethylamine may depend on social experience, on dominance. So if you look at animals that are wild type, the dominant animals, which are shown in dark blue, show perhaps a mild preference for trimethylamine in our assay, but in fact we find that the subordinate animals seem to be averse. And in fact, when you mix the data, we've always seen something very strange with trimethylamine. We've always noticed, we've been doing it for years, we always notice that the data are quite variable. And so this is one explanation, is that maybe it has to do with dominance. And so if it does, there's some very interesting stuff going on here. It's possible, likely then, that there is some learning going on. And we'd like to understand better whether this is true, and if there is learning going on, where is that occurring? Okay, and then the last thing that we're doing is indicated by some of my previous slides, is that we're now using the TARS as a launching pad, as kind of a hook into addressing these very basic questions in olfactory behavior, which is to ask, what is the contribution of individual receptors to both detection, sensitivity, and discrimination? How many TARs are required for the animals to distinguish however many amines you want to test them with? Which TARs are required, and what must their sensitivity or their response profile be to allow an animal to discriminate odor A from odor B? And how does sensitivity at the molecular level translate to sensitivity at the behavioral level? So we're doing things like characterizing the response properties of a bunch of TARs, which are shown in color here and shown as dose responses here. This is all hypothetical. And the idea then will be that you can also threshold the animals. You can take a wild type animal, test them with a given amine. The idea is that these are the response profiles of these TARs to a particular chemical, let's say phenylethylamine. A wild type animal will have some sensitivity, which will be dictated and will somehow relate to the sensitivities of the receptors the animal has. If you delete, let's say, TAR4, the question is, what happens to the sensitivity of the animal? Does it default to the sensitivity of one of these other receptors? If you were to, for example, remove the second best receptor for phenylethylamine, will it change the threshold of the animal? That is to say, is there some kind of cooperativity among the receptors such that it's not only the best receptor that matters, but you also need input from the next best receptor? And how different are these sensitivities from each other to have an effect? So there's all sorts of interesting questions to ask here. Of course, you could remove the entire gene cluster and you will affect the sensitivity of the animals. I've already shown you that. All right, so for right now I said that our model is TAR3 and TAR4. We can record from these glomeruli in vivo. And what we can see is that for phenylethylamine, no surprise, TAR4 is more sensitive than TAR3. And you can see that in this plot. For isopentylamine, the sensitivities are closer to each other, although TAR3 seems to be slightly more sensitive than TAR4. And so what we're doing now in the lab is that we are carefully thresholding animals in which TAR4 is gone, in which TAR3 is gone, and then ultimately we will do animals in which both are gone. But we haven't generated those animals yet. Our preliminary data show that tar-4 deletion animals have deficits in detecting phenylethylamine. I've shown you that already. And it seems to be that deletion of tar-4 does not have a dramatic effect on sensitivity to isopentylamine. The more interesting experiment will be to look at deletion tar-3 animals to see it affect isopentylamine sensitivity. So this approach will allow us for the first time to genetically dissect and to tease out what the contribution of individual genes are to setting detection thresholds in a mammal. And ultimately, at some point, we will also start to look at discrimination as well. And then more broadly, the lab is interested in defining, well, what is the function of this segregation of afferents? And does this mean that downstream circuits are preferentially getting input from these different regions of the bulb? That's something that we don't know. But we can certainly address that using viral tracing. We'd also like to know if the TARs are special and that they project information to a particular region of the brain. Our methods also allow us to partner with people who are doing in vivo recordings in awake animals, and we can now for the first time record from neurons associated with the glomerulus that we know the animals are using to do a particular task. If the animals require tar-4 to detect the stimulus and to discriminate among certain sets of chemicals, it is very likely that the information that you're recording through your electrode is being used by the animal to do the task. And that's something that will be very new to the field. And so we look forward to doing experiments like that. In addition, I have a long-standing interest in understanding how the circuits and this domain structure develops. And there are some genetic experiments that I've planned to get at some of those issues as well. So the folks who did a lot of this work are shown here. Adam Dewan is a very talented, tireless postdoc who did all of the behavioral analysis that I've shown. And some of the behavioral analysis was done with a new postdoc, Annika Sishi, who's from Mark Speer's lab in Germany. Rodrigo Pacifico was the very brave graduate student who took on this TAR project when he first joined my lab. We didn't have any of these reagents, and he was able to very quickly make all of these strains of mice that I showed you and characterize the projections of the TARs. Jingji Zhang is a postdoc who did electrophysiological recordings. They were supported by a host of undergraduates and technicians. The optogenetics work was done by Matt Smear, who's now at University of Oregon, and he worked in Dima Rinberg's lab. Dima is now at NYU. And I should thank David Furster for helping out with the video tracking. The real heroes here who did predator cat urine from the Philadelphia Zoo, and of course my funding sources. So thank you very much for your attention. I'll take any questions if you have them. Yep.
The issue of, first of all, nice one. But a question about the issue of learning versus innate components to a lot of these odor representations. I mean, is it the case that if you take naive animals, like for the social odorants, I understand because they're reared in groups and whatever, but for things like predator odors, right? I mean, it's innate, right? I mean, they don't have exposure to any threat associated with these odors.
So there's very good evidence that they have, they're exposed to trimethylamine because there's a lot of it in the cage. They may not be exposed to high concentrations of phenylethylamine. The question is, do they generalize trimethylamine to the other inputs? It's very difficult to show that something is innate, right? And I think the best chance that we have to get at the real question is to do this rewiring experiment. We did try to do this with pups, for example. P9 pups show the same aversion to phenylethylamine. However, I will note they also show a very strong preference to orient towards home cage odor. So they've already learned something very early. And so that experiment is off the table. It's not telling you anything about innateness. It's only telling you about what the animals are doing. So I think it's a very sticky issue, this innate business. So the long and short of it is I'm not entirely sure that the TARs are really there only to mediate innate behaviors. What I really think is going on is that the TARs are there because they're the kind of receptor that you need to get high sensitivity to amines. And whatever the system does with that information maybe it's hardwired, maybe it's flexible, but really you just want to get the information into the system, and maybe these receptors are the ones that you need to do that with appreciable sensitivity. That's one idea. Yep.
Also I want to reiterate, lovely talk.
Thank you.
Curious about looking at your experimental setup with the head restrained animals that are getting odor exposure. So the concentrations that you're showing us in your sort of dose response curves, that's in the air that the animals are sniffing. And so that's into an environment where there's dispersion and whatnot. So you have some sense of what concentration is actually reaching the odor receptors.
Yeah, so this is a very good question. And again, broad strokes, I washed over all this. In the head-fixed behavior, we have very good control over the concentration. The distance between the animal's nose and the odor is the same. And we're very careful to deliver the same concentrations. And so the sensitivity curves that we get are very consistent, actually, if you do things, if you're very careful. It turns out that the sensitivities that we see in the behavioral assay for thresholding, those sensitivities are strikingly close to the sensitivities that you see in the patch clamp assay. So for example, the animal's thresholds for phenylethylamine are within a factor of 2 of the thresholds that you see in the patch clamp assay when you're stimulating the neurons with aqueous solutions of phenylethylamine. That may just be a happy accident. It's something that we'll have to flesh out. So that's something that we've observed. What is much more difficult to know is in the aversion assay, when you put a dish in the cage, whether how those concentrations relate to the thresholding. We just don't know. And so we would need to do a different assay to control the odor better, but we don't know how to do that for aversion. We've been racking our brains to think about how to do it, and we just don't know. So it's a very good question, and we don't have a good answer.
Let's thank Paul for a fine talk, and maybe stay after for other questions.
Good. Thank you.
That's great.
Okay, I was almost on time. I think we started like 5 minutes late, yeah.
I was curious about the